the lecture. So the Michael Potter Cardiovascular Genetics Lectureship was actually established in 2006. So it actually predates our 2007 entry into the big world of complex genetics. Michael Potter served for 23 years. He was president and chief executive officer of Cognos, and I think most of you know about Cognos, the multi-billion-dollar global company he founded. He was born in London. Michael isn't with us today, is he? Michael was born in London, and he arrived in Canada at the age of 70. He's a graduate of the Royal Military College and the University of British Columbia, where he did his MSc in physics. Michael and his family have a real affinity for the outdoors. Michael is an experienced boater and offshore. Oops, I'm destroying your lecture. Sorry about that. One sec. And offshore sailor. So we're very grateful to the Michael Potter family for the opportunity to have this lectureship. We haven't had one for about 3 years because of the interruption of COVID. So we're really delighted today to introduce Dr. Tim Assimes, who is an associate professor of medicine at Stanford University Division of Cardiovascular Medicine. So he was born and raised in Montreal, medical degree at McGill. He subsequently pursued training in surgery, but found that surgery wasn't his calling and switched to medicine. I'm sorry, Dr. Masana, but those things happen. He completed his residency in internal medicine and a master's degree in epidemiology and biostatistics at McGill, and then moved to Stanford to pursue fellowship training in cardiology. And concurrently, he completed a PhD in epidemiology and biostatistics. So this is a perfect blend: medicine, cardiology, and of course, epi and biostatistics. And you'll see that today when he presents his findings. So he currently devotes most of his time to advanced population-based research on the molecular determinants of coronary artery disease and its comorbidities, including dyslipidemia, smoking, diabetes, obesity, and hypertension. So he also teaches general adult cardiology as well as echocardiography to medical students, residents, and cardiology fellows, and who we hope that at least some of them will pursue this avenue of research. Tim, welcome.
All right. Can you hear me? Okay. Good. All right. So thank you. Thank you to the organizing committee for selecting me to give this lecture, particularly Ruth and Alex, who have been partners in crime for many years through the Cardium C4D Consortium. I'd also like to thank Michael Potter and family for making the endowment possible. It's wonderful to be here, especially in the early fall. Greenery is at its maximum, contrast to the dry state of affairs on the West Coast. So it's something I really miss, the 4 seasons here. That and good community hockey rinks. We don't have those in the Bay Area. No better place to give a talk on GWAS for coronary disease, because the era for GWAS started here. And I would imagine that most of you have heard about this initial publication where Ruth led, Alex and Bob Roberts were co-authors in Science in 2007. This was the first GWAS for CAD and led to the discovery of the first common genetic variants influencing risk of CAD. There was a second paper by the DCODE group that followed. They were published at the same time, but I will note that Ruth managed to submit 1 week earlier, 9 days earlier. So from that perspective, it is the first. It's actually, if you do the math, it's 16 plus years, but that didn't have a good ring to it for my title. So it's 15 plus for me. One thing to also note on the right side of the slide is from the get-go, they used to do the international level of collaboration that was necessary to make the discovery. You worked with people from UT Houston, UT Dallas, and in Europe. And this collaborative spirit was necessary to make sure the findings were convincingly replicated off the bat. And it continues to this date, not only for replication, but just for the benefit of bringing together many curious and capable scientists to make sense of the GWAS data. Wide variety of individuals attending, faculty, nurses, undergrad, and translational, basic scientists. So I thought in order to try to get a lot of you on the same page, I'd start off with a little primer for 5-10 minutes about genetics. I think the sort of key facts that you need to understand the rest of my talk, I hope. So just summarizing, we have a code, a DNA code, 3.1 billion bases long, divided in 23 chromosomes. We get one copy from mom, one copy from dad, and any two individuals in the room will be 99.99% the same, but that 0.1% makes you unique and makes a big difference, not only externally, but internally in terms of your predisposition to disease. Some common definitions you may be aware of already. A locus is a specific region of the chromosome, and a polymorphism is when in that region you see more than one version of the locus due to a new mutation. And so those different versions are called alleles. Sometimes we might refer to an allele as susceptibility allele if it predisposes disease, or protective if it does not. And here you can see a region of the chromosome, your base pair is coming along. There's only one difference. And we'll talk about the different types of mutations right now. That one difference is what we refer to as single base substitution. And it's referred to as a single nucleotide polymorphism, commonly known as a SNP. And that basically makes up for vast majority of the mutations that we see in the human genome. The one thing is to remember there's 4 bases, but when you see a polymorphism, you'll almost always see just one of the other bases and not all 3. Sometimes you'll see all 3, but very rarely. Most of the polymorphisms, it's a pair of bases that are involved, not all 4. So most of these do nothing in terms of biologically speaking. A very small number will have some major disruptions to the function of the protein encoded by the gene. A few will just change an amino acid, and many, a vast majority have just very subtle effects on gene expression and therefore the amount of protein. There are more complicated mutations known as insertion deletions and large-scale structural variants involving a large fraction of the chromosome, but we won't talk about those because they're not that common, at least not in complex disease. So then if we think about a SNP and we look across 5 individuals and 10 chromosomes, we can define, allele frequencies based on what we observe in those 5 individuals at that specific region. In this case, you can see that there's, in one specific location, 3 C alleles and 7 G. And the C is therefore the minor allele at a frequency of 30%. And if that C increases risk, we would refer to that as the high risk allele as well. Determine what's going on, what do we see? There are 2 different ways. Sequencing, that's the longer, more expensive, but getting cheaper way, which is basically starting from scratch. You get a piece of DNA and you figure out every single base in that small piece, and then you get another piece and you tile everything together. But another way is once you've seen variation, once you've documented variation of a specific location, instead of trying to figure out every base pair, you could just land a probe and say, well, what's going on right after my probe? And that's basically the way the genotyping arrays work. They really only needed to figure out what was going on in regions that they had already documented polymorphisms, and some of the arrays now are going upwards of 2 to 3 million SNPs. And a large fraction of the GWAS data that we've generated have used arrays, and now we're starting to move into an era where more and more of the data being generated is actually sequencing, whole sequencing. The Human Genome Project was the first attempt to actually figure out that code, 3.1 billion base pairs. It was a small number of individuals, I think about 8, so 16 chromosomes. And in addition to figuring out that first draft code, we also, because there were more than one chromosome, we were able to identify the first map of genetic variation, which included about 1.5 million SNPs. that had a frequency of higher than 1% of the expected 11 million. Now you fast forward, 15 years later, and you have large-scale sequencing efforts. And here's one of them that I've been involved in, funded by the NHBI. It's called TopMed. It is focused on heart, lung, blood diseases, but they've now sequenced about 150,000. But in the 54,000, the first 54,000, you can see that we're starting to document many variants. There's millions of very rare variants that we've never seen before. A large fraction of them were never seen before or only seen on one chromosome. But what's happening at any given individual, one individual, you, me, we have 3.8 million variants, of which 3.6 million of them are SNPs. And most of them are common. Only a small fraction, about 200,000, are rare with a frequency of less than 0.5%. But one more primer slide, and then we'll go on to the fun stuff. So there is a difference between monogenic and polygenic disease. So monogenic tends to be rare. It tends to travel very obviously in families. You see the pedigree on the right. So there's a very high penetrance. One base pair equals disease. The environment often doesn't influence the outcome too much. You can see multiple affecteds. And those variants or variant typically tends to be pretty rare, but the vast majority of the diseases we take care of as cardiologists are actually not that, they're polygenic. So a very large number of those common SNPs are involved to affect the probability of disease. And all of those effects, as you'll see in a moment, are relatively subtle, with few exceptions. For CAD, for example, the vast majority of what we see is polygenic. Perhaps the single exception is many of us are familiar with the monogenic presentations of familial hypercholesterolemia with individuals presenting in their 20s and 30s with coronary disease and often sky-high LDL. So those are due to really bad mutations in LDL, ApoB, and PCSK9. Was preceded with at least 2 decades worth of failed attempts to identify genetic variations in genes responsible for complex traits through linkage studies. And several population geneticists were thinking about this, and they were anticipating, well, we're going to be documenting all this genetic variation across the entire genome. Do we continue to do what we've done or do something differently? And specifically, Drs. Neil Risch and Maritangas had done a lot of thinking about this as genetic epidemiologists and the implications. And they published on the left side, you see, that's just a letter in Science in 1996, 10 years before the first GWAS was published. And they were arguing that we need to move away from the family studies, and we gain a tremendous amount of power if we analyze by association rather than by linkage. And so we liberate ourselves from the need to recruit, the challenging need to recruit families, and they also knew that the effect sizes are bound to be modest. It's interesting that in this table, if you look carefully on the left side there, they document power for odds ratios of 1.5 to 4. And you'll see in just a moment, 1.5 was would have been a dream for CAD. So essentially, genetic studies became the purview of epidemiologists because the most efficient and powerful study design, as you can see on the right, was just to do a case control study and look at cases, look at the frequency of the SNPs in the cases and at controls, and you see the difference. You correct for multiple testing, 1 million independent tests at 10 to the minus 8 p-value, and that simple design has gone a long way, actually. So here's the first key question answered. So CAD is heritable to a substantial degree, and the heritability is about the same across all populations. So first of all, I would define what is heritability for those of you who may not know. It's the overall proportion of, the overall proportion of the variation of a health trait that can be attributed to genetics as opposed to something else, which is typically environment. And before the modern era, the genome era, there was a variety of ways to sort of estimate how genetic a disease was, and perhaps the most embraced and practical way to do that was twin studies. basically for a disease outcome, you'd compare how often a pair of identical twins both have disease to how often the fraternal twins both have disease. So the more genetic a disease is, then the bigger that difference is. So one important assumption and why this design was embraced was that the environments were equal for a set of identical twins versus a set of fraternal twins. Some people tried to challenge that, I think, but for the most part, people feel like they're close enough. So they cancel each other out, and then basically you can calculate heritability by taking the Falconer's formula, which is the difference of the correlation between monozygotic and dizygotic twins times 2. Don't ask me why that's 2. I don't have to go back to the derivation of the equation. The other thing, so in these, the big study for CAD was published in the New England Medicine Journal in '94, and Neil Risch was and Kathleen Maricangos were also lead authors on that paper, and they estimated that the heritability was 40 to 60%, males versus females. That was sort of replicated a few years later in the Danish cohort on the right, as you can see. The other thing is, through some additional analyses, they estimated about half of that heritability was due to traditional risk factors. And these studies were obviously, since they were in Northern Europe, restricted to Europeans. So in the modern genome era, when you have all these SNPs genotyped, you have also new approaches to be able to estimate heritability. One of them is called GREML, or genomic relatedness restricted maximum likelihood. And here you use unrelated individuals who are assumed to, so unrelated individuals are assumed to vary. They have some genetic similarity, even though they're unrelated. It's typically 2.5% or less of their genome is shared between any two individuals. And that might be due to like a very distant common ancestor, or maybe it's the same mutation appearing in different places multiple times. And so in that, so you can take a set of individuals who have been genotyped very densely and just look at the similarity in the pairs and say, okay, here's all the pairs that are 1% similar versus here's all the pairs that are 1.5% the same genetically, and then 2%. And then you look at that slope and you compare it to the slope of how similar the phenotype is. And so the tighter that connection is, the more heritable the trait is. And then the environment is independent, because these are unrelated individuals that are not, they don't know each other. So using these approaches, we were able to estimate heritability and across 4 different populations. So Europeans, Africans with very little white admixture, Japanese, and Hispanics with maximal native indigenous ancestry. And we got basically heritabilities are in the 30 to 40% range, highly overlapping, suggesting that the heritability is the same. It's a little bit lower than what you see in twin studies, but there's some of the variants we don't identify gene-gene and gene-environment interactions with this method. The other thing is, I should mention, is the twin studies were that the trait was death from CAD, which is more heritable than just any CAD, for sure. So that 40 to 60% is on the upper limit. It includes gene environment, gene-gene interaction, and it's a more severe phenotype. So these are starting to make sense. They're congruent. Plus, if we had more rare variants, would be higher, I think. And it's heritable, basically. At least a third of the variance of CAD is related to genes. So the next one is, when you look at the genetic architecture of CAD, you find that it's complex, much more complex than originally thought. I mentioned early on that Neil Risch had done those power calculations, and they were hoping for 1.5 to 4. He was actually, I had spoken to him for a few years. He was still at Stanford when I first arrived, and he's hoping that like there would be 5 to 10 SNPs that had a frequency that was, pretty high, and an odds ratio of like 1.5 to 3, and that would explain a large fraction of the genetic variance with just those 5 or 6 and then that didn't pan out for almost any disease. So think about BRCA1, for example. Those are high, common and high penetrant in the context of complex disease, or ApoE for Alzheimer's. So we were expecting to find 5 or 10 of those for almost any phenotype, but also CAD. we didn't. It's a little, and I can show you what we have right now based on the latest 2 studies that were published. But before I do that, I just want to give a little bit of a historical context. So 2007, the initial publications. And then in 2011, there was some consortia forming. This is, I think, where we basically met. The Cardiome Consortium and the C4D Consortium published in 2011. Again, mostly European. In 2012, we had 2 GWASs in Japanese and Chinese. In 2013 and 15, subsequent GWASs for when the Cardiome and C4D joined. These were largely, up until recently, population of European, South Asian, and East Asian descent. In fact, a majority were European. There's this European bias that we'll talk about more later on as well. And then in 2017, the biobank sort of era expanded dramatically when the UK Biobank genetic data came online and the world started using that. But of course, it was 97% white, so it didn't increase diversity. In 2017, it's around the same time I started also working on the Millen Veteran program, which at least initially offered a smaller number, but a much more diverse data set, with about 20% of the enrollees being African-American and about 7 to 8% being Hispanic. And then in 2020, there was a Biobank Japan published their big GWAS. And then all of these groups sort of worked both in parallel and together in the last 3 years to produce the last round of GWAS that you see here. I was fortunate enough to be in a position to lead the MVP-centric study on the left, while the Cardiogram plus C4D study was led by Adam Butterworth and others on the right. And these studies brought together almost 250,000 cases. One thing to remember is when we do GWAS for coronary disease, we only adjust for age and sex. Actually, sometimes you don't even adjust for age. So the results of your association analysis, you would expect, you would hope that a subset of them would be pointing to traditional risk factors. And that's actually a benefit of not adjusting for traditional risk factors, just adjusting very basic sex. And often also in case control studies, it's hard to actually document risk factors reliably. These 2 studies published last year in August and December in Nature Medicine, Nature Genetics. Ours on the left reported 95 loci, new loci, and added to the 210 that were there already. The one led by Adam, another 30, there was some overlap. So roughly now we're at 320 independent regions of the genome that have reached that sort of magical threshold, 10 to the minus 8 that I mentioned earlier on. Not magical, but high confidence. Our study we were the first to report regions that reach genome-wide significance for African and Hispanic populations, as well as 6 regions on the X chromosome. And going back to the original sort of key question answered, it's complex. These are the hits, not all 320, I think about 200. And you can see the effect sizes are nowhere near 4 or 2, even 1.3, and these are kind of rare. This is actually 9p21, that's the highest one. Everything else is down here below 1.1. So 10%, 5%, 3% increase. And here there's some color coding with the new ones being in red, and you can see the new ones are all invariably have very modest effect sizes. So we've basically, I think, all common variants with high effect sizes for coronary disease have already been discovered. We're working here in the trenches to fill this space in. We're working, we won't quit. This is a data set that it's unpublished, but it's Cardiome plus C4D and MVP together. And we have now, and others who have come in, and now we have over 600,000 cases. We're probably going to freeze this meta-analysis soon, but we've done some preliminary analysis. And sure enough, now we go at over 500 loci, and you can see all the novel ones. They all have very, very modest effect sizes here in the 1.02, 1.03 range. So when I was doing my epidemiology training, like they were telling me this is all confounding. It's not real. This is residual confounding. These are all real though. It's taken 600,000 cases and 2.5 million controls to be confident of that. So it's very complex and nothing in the range of the 1.5 to 4. And here I sort of like in red, we'll see all the regions of the genome where the novel hits exist, compared to in blue, the previously known. So how do we make sense of these results? It seems like the larger the sample size, the more discoveries will be made. Sometimes it feels like maybe eventually the entire genome will be implicated. And in fact, for height, it has.
19,000.
Not 19,000 what?
19,000 loci. That's what you guys are.
Oh, yes. Thank you. I will do that. I'll tell my team and see what they say. So I'll give you a model in just a second. So these 500 loci are only explaining about 1/5 of the genetic variants for coronary disease. So there's still a lot left to be explained. And so I'd like this model. We go from polygenic to omnigenic. I like this for a couple of reasons. One, it simplifies a complex situation. And second, there's an implicit bias of mine. The senior author here is Jonathan Pritchard, a well-known human population geneticist, also happens to be a colleague and lives across the street from me, has kids the same age as mine. They grew up together, they're friends, and they're still friends and they're kind to each other. And so I give them the benefit of the doubt. All kidding aside, the model is based on control of gene expression, either directly cis or indirectly trans. And control of gene expression is something that Jonathan has studied for the last 1 or 2 decades. And the idea here is genetic variation perturbs gene expression and therefore the quantity of protein made in the cell in general. So what I want to emphasize here is that it's not all genes that are involved, but I think there was a misunderstanding initially, and Jonathan was telling me this, like, oh, it's all genes can be involved within the cell types that are relevant to disease. Because not all cell types are expressing all genes. So the idea is that like cell types that are relevant to CAD, then that's where you would look, where most of the heritability would be. So the model starts with defining a core set of genes. You can see those in the black as the set of genes that have direct influences on the trait. And they're not mediated through regulation of other genes. However, because the presence of many genes, there are many gene regulatory networks, you can see them there that overlap in a cell to a certain degree. The core genes are likely to be influenced by hundreds of peripheral genes and the genetic variation of those peripheral genes. So cis regulatory variants, direct variants affecting expression of peripheral genes, are also weak indirect effectors of the core genes. And that this way, you could see how a large fraction of the genetic variation of the heritability could be explained in those peripheral genes and not necessarily the core genes. There could be some peripheral master regulators that would, if they regulate the core genes all in the same direction, would be strong GWAS hits. Complex. Next question. The genetic drivers across populations are all likely all the same as long as a causal genetic variation exists. All right, so I mentioned to you earlier on that in our GWAS, we were able to find we were able to get high enough sample size and power to identify the first hits for Hispanic and African populations, or 8 of them. They were all known in Europeans. Like, these are just the locus zoom plots. These were not surprising. You can see they're listed here on the left. And just to extend these findings to this larger GWAS, the 600 case one that's coming, 600,000 case one, 71 regions in that multi-ancestry GWAS have reached significance in non-Europeans, and 69 of those 71 have been identified already in Europeans. So it's not as if we're going to go into some other population and see a totally different genetic disease. It's the same, at least based on our largest sample right now, which is Europeans. So if we were increasing, if we had very large sample sizes for all ancestries, we would expect eventually all the same genomic regions to be implicated in every group. That's what we expect. except if genetic variation doesn't exist for a particular locus in a particular ancestral group. And you may have noticed here is that 9p21 isn't on this list, even though it was the top hit in Europeans, one of the top hits in South Asians, and a top hit in East Asians. So actually, this is a history of 9p21 after the initial discovery, widely replicated in European populations, then replicated in South Asians and Chinese, Japanese, with effect sizes 1.2 to 1.4, so pretty large in the grand scheme of things. And you would think that all you needed is 1,000 or 2,000 cases in other populations to discover it if the effect sizes were the same. But multiple attempts in African Americans and Hispanics wasn't able to identify the 9p21 locus. And this is something we sorted out in the paper, is we have a large enough sample of African Americans. We also didn't see, with 17,000 cases, we still didn't see an obvious signal. We saw a little blip in that region, and then we asked ourselves, well, if we see it's very strong in whites, and African Americans have some admixture with whites, they have about 20% that's been introduced since they were brought to America as slaves through mating with whites. So 20% of their genome, and you can actually, with a high density genotyping now, you could basically paint the chromosome. You may have, like, if you've done ancestry.com, you may have gotten your own painting. And in this case, it's white versus African, or European versus African, and you can find the regions. So we did this sort of local ancestry aware analysis. And what we found is, like, if you look at the African Americans that have in this region in 9p21, their chromosomes are coming from Africa, both of them. Despite having a huge number of cases, there's no signal. That's in the top left, right here. But then you look at a much smaller number of African Americans that have at least one chromosome that's white, and you see a pretty strong signal. It's not genome-wide significance, but this is looking exactly like the signal we see in whites. In Hispanics, we did the same thing. We couldn't quite reach genome-wide significance. We removed Hispanics that had African ancestry in that region, and the signal became genome-wide significant. And basically, the causal SNPs are not in the, they're present in every population except for African populations, especially West African, which happens to be the main region from where African Americans came to Africa from. The genomic regions identified to date appear to be mediating risks through traditional risk factors. This is back in 2017, about 60 hits had been identified back then. And if you compare the results for CAD outcome to the results for traditional risk factors, you could see links here. There's strong links. There's a lot of links with the lipids here in red, blue, I don't know what color this is, gray. There were less so with the obesity, insulin resistance, diabetes axis here. That was fewer of those loci, and a lot of people are wondering why that's the case. Hypertension was very strong too. When we did our GWAS, much larger numbers, we started to fill in those gaps. Still, 50 to 60% of our novel loci are tracking with traditional risk factors. Still a large fraction of those are lipids and blood pressure, but now we're starting to see more of those related to diabetes, obesity, and even substance use disorders. So things like the top hit for obesity had not reached genome-wide significance yet, like FTO, some of you know that. The top hit for diabetes had not reached genome-wide significance yet for CAD, now they had. And the top hit for NASH, or insulin resistance, PNPLA3, had not reached genome-wide significance. It did in our study. Another key question. Answered. Polygenic risk scores for CAD and related traits improve risk stratification for CAD. So there's clinical validity. There's lots of debate on clinical utility, but there's definitely lots of clinical validity for polygenic risk scores. A little bit of a primer again. How do you create a score? Well, once you have a set of SNPs that are associated with the disease, and you have the weights from your regression models, you basically just add up the number of high-risk alleles, and at each SNP, you multiply it by the weight, and that's your score. And you end up with a normally distributed score here. And those who are at higher risk are over here all the way on the right. We can compare these to the rest. Another way to compare these scores is to just look at increasing risk per one standard deviation shifting, no matter where you are in the distribution. So we often hear this, what is the increased risk per standard deviation increase in the polygenic risk score? And the larger that is, the better the score. And basically, we're working on more elaborate ways to figure out the best weights. And so this describes the first part here where you have the GWAS, you have your list of SNPs, you calculate your PRS, there's some tuning parameters here, then you train them on independent cohorts, and then finally you validate. And that's where you end up, this is where you determine your AUC, your p-values, your hazard ratios, and all that stuff. And then that one standard deviation increase in risk highly correlates with the delta AUC, which is typically small. So some properties, these are highly reproducible, the scores in general, not just for CAD, for most traits that have been studied. They can explain up to a quarter of the heritability of health traits to date altogether. They independently predict adverse outcomes, and they work best when they're used in the same populations they were developed, as we'll see in a minute. They are improving over time. They're definitely improving. I'll show you some data on that. More SNPs, and there are better ways to estimate these weights. And you just have to do the test once, sequence or genotype, and you should be able to create many scores. And then you can imagine a day when you would just integrate this score. It's like a biomarker. It's like anything else into your risk calculators. And the idea is you just modify your 5 or 10-year risk and modify the type of well-known primary prevention strategies that you would recommend depending on that risk, 5, 10, 30 years. And I mentioned to you earlier on that about half of the individual SNPs are tracking with disease. Well, the PRS is even stronger with traditional risk factors, I should say. It's the same thing for the polygenic risk score. And you can see here that we did this sort of phenome-wide association study where we checked the polygenic risk score for CAD in people who did not have CAD, and the top signals were hyperlipidemia and hypertension. But in our study we were the first to actually be able to show that it also tracks with diabetes. It also tracks with substance use, tobacco-related predisposition, and then pretty incredibly, family history, not only of CAD, family history of all the risk factors of CAD. So all of these signals here in your polygenic risk score, you do pick up family history signals. This is the Achilles heel of polygenic risk scores. That because we have most of the GWAS in Europeans, they don't work as well in other populations. The more genetically dissimilar the population is from the base GWAS, the worse those scores do. And probably the population that suffers the most is the African population. So you can see here that most of the GWAS done in European, even though the Europeans are only a small, less than whatever, 15, 20% of the entire population. And then you can see benchmarking here for quantitative traits. Here's what the polygenic risk score does in Europeans when developed in Europeans, and then it just sort of deteriorates as you go further and further away in other populations, ancestral populations. And that carries through. This was all quantitative traits here on the right. Oh, I'm sorry, you're not seeing my. And this is disease outcomes. And the main reason behind all of this is that the correlation structure of these SNPs. SNPs are introduced in the population in a specific individual. We have our own SNPs. All of us have about 30 or 40 new ones that none of our parents have. And they're introduced in populations, and they're introduced with a background set of other mutations, and they travel together. They're blocks. We call them linkage disequilibrium blocks, and they will not separate and become independent until there's recombination between them. And that doesn't happen too often. It takes multiple generations. So the foundation of this lack of transferability of scores is that those correlation structures are different Europeans versus Africans versus other populations. And so one way to try to overcome that is to actually do more GWAS in those non-European ancestral groups, use that information to improve polygenic risk scores across the board. This is a paper that we published. Actually, it was led by Anurag Patel and Amit Khera, but the validation was largely done. It was published in Nature Medicine a few months ago. It was done largely in the Million Veteran Program that my team helped. And actually, just here you see a progression of improvement in the hazard ratio per odds ratio over the last 7 or 8 years, different ways to create these scores. And then finally, this last one at the top here, the red ones, are actually multi-polygenic scores, polygenic scores for CAD and risk factors for CAD combined. But and things look like they're getting about equal for multiple populations, but you can see that the African population is still lagging behind in terms of hazard ratios. They're down to 1.25 on the left. And while things look good here for Hispanics, we were able to show that you have to be careful with Hispanics too. There's about 10% of them that have high African American ancestry, and those scores break down. This is a short letter we published in Circulation, I think a year or 2 years ago. And you can see this is the group here on the bottom. In brown, you see there's 3 clusters. We clustered the Hispanics in 3, and one of them clearly has almost 50% African ancestry. And they're if you're not aware of that, you're not going to help them with the score as much as you would if you knew. All right, so next question. A good fraction of the loci that we've identified for CAD promote development of atherosclerosis rather than plaque rupture or thrombosis. So there are 3 recent maybe hurry up. 3 recent observations, 3 studies, 2 of them that I was involved in, led. Actually, this one is not published yet, but it is a building off first iteration. We did present this last year at AHA, working on submitting a manuscript, but we're able to, in the Million Veteran Program, the nice thing is that there is a registry. It's a quality assurance registry of the outcomes for all coronary angiograms done across the nation. It's been mandated since 2009, introduced in 2005. And so we have burden of disease, number of vessels, all sorts of information in that registry. And we're able to cross that with the participants in MVP, 65,000 here. And we basically did a GWAS, a burden of disease as estimated by the number of vessels that have high grade lesions. So 0, 1, it was 0 non-obstructive, 1, 2, 3 left main. Nothing more fancy like scores, but like Gensini scores. We could only do that for now. And then what we've identified is 46 regions that have reached genome-wide significance in different race ethnic groups, most of them in Europeans, but all but two are already known from the clinical CAD case control studies. And most of these were also discovered initially a long time ago in the first 5 or 6 years of the GWAS era, again, case control. So burden rather than plaque rupture here, it suggests that many of the loci are operating through burden rather than plaque rupture. Another study that we published recently is in the PhD study, is an autopsy study of adolescents and young adults, 15 to 35 years old, who died of reasons that are completely unrelated to coronary disease, accidents, suicides, homicides. And their right coronary was dissected, and they documented exactly how much lesions there were. Coronary, basically there was fatty streaks and some raised lesions in a subset of individuals, and we were able to, so you can see here the progression of atherosclerosis here. We're operating in this region here, which is very uncomplicated. There's no plaque rupture, there's no thrombosis, and then we asked ourselves, well, what are the polygenic risk scores doing? Are they correlating with this very early lesion that it's very clean. There's no thrombosis or plaque rupture there. And in this cohort of only 560 subjects, about a third had some raised lesions. We found that the contemporary polygenic risk scores correlate with that very, very early lesion. So that's another reason why I think most of these are plaque promoting loci. And the last observation is here. This is a study looking at incident versus recurrent events. So polygenic risk scores. And what we see is a muting of how well those scores do. The same score does pretty well on the left side. For incident events, as soon as you go to the recurrent event, it's all muted. It's all much lower odds ratios. And this is because then you have a set of individuals that all have coronary disease, and now you're asking, well, the recurrent event, you would think that part of that would be thrombosis, plaque rupture genes, but the fact that it's muting a lot to me means that they're still mostly plaque predisposing genes. Is sex differences. We see sex differences in the burden of CAD appear to have a widespread genetic basis to them, and I think there's still a lot of work to be done here. But we do know this is just the epidemiology paper published a few years ago showing that after 55, we know that the burden on males is much heavier in terms of coronary disease, even though the burden for stroke is higher. In females, it's the line in blue shows that like the lifetime prevalence is basically double when adjusting for competing risks. So one of those two studies that was published, not ours, but the other one, did a sex interaction analysis in about 77,000 cases, looking for differences of effect sizes of the loci for CAD in males versus females. And this becomes a problematic, even more testing. You have to be careful with you're correcting properly. But they did find 10 that passed that threshold of multiple testing. And the interesting thing here is 9 out of 10 have effect sizes in males that are stronger than females. So a vast majority of effects of loci, 9 out of 10 of the loci are showing a larger effect size on males, and that may be one reason why there's a larger burden in males versus females. And then another study published in Circ GPM, led by a colleague of ours who's an MVP investigator, but this is in the UK Biobank. They looked at the polygenic risk scores, and they looked for interaction for the CAD risk scores. And they also found that the risk scores statistically have a higher, per standard deviation, higher effect on males versus females. Where do we go from here in the next 15 years? Well, we still have to continue to identify genetic variants, 19,000 maybe or more, or genes. Importantly, as I hope I've emphasized throughout the talk, in understudied populations in particular, but also across the board, we have to start documenting associations and all of the rare variants with the different complex traits, including CAD. And this gets very complicated. Gene and gene environment interactions. There is definitely some heritability there that we need to uncover. Some of you may have seen a recent paper published, Lipids and CAD in Cell, from the Decode group. I think it was just published 2-3 weeks ago, that tackles this specific issue. In the meantime, what do we do? I really feel like there's more than enough evidence that having genetic data in general, like the whole genome, ideally, possibly available for our patients. Like, I think we have more than enough data. I just don't know why integrated healthcare systems haven't sort of triggered the infrastructure where you can at least generate the data. Don't have to use it. The patient can use it when they want to, but let's not waste time. Let's just generate the data, and then in the background, we can use it when the patient would like to use it, and where we're comfortable, we've done the research that it's reliable, including polygenic risk scores. And then, of course, there's this very, very difficult job of translating hundreds and thousands of hits in these gene regulatory networks, trying to figure out what the core genes are that you can therapeutically target, either with small molecules, antibodies, or gene editing. to hopefully reduce the prevalence of disease and improve outcomes for CAD. That I think is going to take. I mean, we have some agents that are coming out that are being tested, but it's next 15 years will be very interesting from that perspective. Maybe if we find a few therapies, we won't have to do the other two stuff because we'll just wipe out coronary disease, and then we can all retire early too. But, just quick special thanks to there's a lot of people I can thank here. On the bottom are the either postdocs or ex-postdocs that helped with the papers that we published last year of MVP, research staff, and two people that have influenced my life quite a bit. One is Tom, who basically asked me, I think in 2003, he's like, how about you do some genetic epidemiology instead of pharmacoepidemiology? I said, okay, I'll try that. And I've been doing that ever since. And Phil, who in 2016 said, you know, I somehow wrapped myself, I got so involved in MVP, like almost by accident. Do you want to help me? And I was like, sure, I'd be very happy to help you given that wonderful resource. So I'll stop there and take questions.
An amazing overview and really exciting new data you're showing. So you talked about some of these genetic variants having a much stronger effect in men than women. Did we actually screw up in the way we did GWAS studies, knowing we knew full well that, you know, coronary artery disease happened 10 to 15 years earlier in men than women? Yeah. Shouldn't we have perhaps a priori separated men and women and done the genetic studies that way? Because when you correct for, when you adjust for a variable post hoc, it doesn't quite have the same power as doing it a priori. So I'm wondering if perhaps we would have made other discoveries, or have you tried going back to the original data, or and seeing if you repeat the GWAS sort of segregated by sex, if we would find additional loci?
Yeah, I mean, I think there the one thing that in the slide that I showed you that had those differences between males and females, the 10 loci, they were not able to use the entire data set because only a subset of individuals were willing to redo analyses. You know, males and females, every group is limited in terms of their resources. They may have data, but then, so you know, from that perspective, I think it's true. You're right. We should be, as we're gathering data, we should be emphasizing, you know, we'd like to do a stratified analysis up front. That being said, I will say that some are like, okay, we just need larger power. They've prioritized that because the biology is largely the same. But yeah, I think that, and from my perspective, in MVP, we have very few females. The veterans are all males. So in fact, there is 5% females, but there's very few cases of CAD. So I can't even do a stratified analysis in MVP. But yes. Yeah.
And one other question, 9p21, we're still, so where do you think the difference lies in susceptibility to that risk variant, to those risk variants on chromosome 9p21.3?
Yeah.
Do you think it has something to do with Alu sequences?
With
Alu sequences?
Allo sequences?
Yeah, so the Alu element is distributed all over the genome.
Oh, Alu, okay.
But
sorry.
the frequency of Alu elements insertions in African Americans is lower than in Caucasian and Europeans. And I think sequences also are not consistent across ethnic groups. Because I was really stunned at your observation about the Hispanics. Also, we've seen some weird numbers even in Europe with incidence rates on other diseases that sort of diverge among the Spanish population relative to other Europeans.
I haven't done any analysis with Alu, but I think I didn't get into the full details, but I think for us, what provided clarity in the situation was doing this haplotype analysis among African Americans. Like, so in whites, you have like 50 highly correlated SNPs. All those SNPs exist in African Americans. We took the subset that were least correlated with each other. And that's when thing became clear that those haplotypes do not exist in certain populations. It's only basically African-based populations. So I think for us, without going any further than that, that's why we concluded like it's just that genetic variation is not there. Either it was introduced later on in the populations after they left Africa, and then it also must have had some sort of survival advantage too, because it went up to 50% in most populations.
Right. Thanks.
Yeah, that was actually one of my questions, and that is that the 9p21, then because sort of the African ancestry, obviously have quite wide heterogeneity, right? And
Yes.
that the Europeans, are much more, I guess, more homogeneous, and the 9p21 must have been somehow selected out, right, as a survival factor to be so strong at the same time, become a susceptibility gene, right?
Yes.
Yeah.
If it was introduced after the populations left Africa,
Right.
that I don't know how many hundreds of thousands of years ago, but then you have to see that. So to rise to 50% means it must have had some selective advantage. Now, it's not 40 to 50% in all populations. It's a little lower in East Asian and South Asians, but it's still common.
Right.
And I don't know, we've been scratching our heads on that. What is it? What benefit? To rise to that level very quickly is pretty amazing.
Yeah, so this is really striking. So the comment I want to make, and this is about your kind of a final discussion in terms of the use of polygenic scores. And, so we do a lot of work in biomarkers. And so one of the things that is very important is that, a diagnostic tool is useful only if it can actually have some impact on clinical decision-making, about, sort of treatment. So I wonder, so for example, you actually demonstrate very nicely that, it can actually predict, for example, plaque burden, in terms of the, eventual, even recurrence of the disease. So it definitely has very important influence in terms of trajectory of the conditions. The question is, what's the intervention that could actually introduce to actually make, sort of that mitigate, against that risk. So I wondered whether, for example, even using statins and things like that, whether one could actually use these calibrated, sort of a risk, and then look at the potential effect, because some of those patients probably happen to be on statins anyway, whether there is a potential, mitigating effect of statins.
Oh,
of
yes,
those patients,
actually.
so because then,
Yes.
then you can make the connection, right?
So I'm not sure if you're aware of this but there is a pretty well established, I would call it gene PRS effect, which is there's been multiple groups that have published on, I didn't talk about this here, but PRS, those who have high PRS with CAD will benefit from statin use more. Their risk reduction approximates 50%. I suppose those that are lower risk will benefit less. In fact, some of those don't benefit at all. And that is, there's a few groups trying to work on that, including groups at UCSF and Kaiser that I've worked with. It's been replicated, and it's a very interesting gene environment.
Right.
Yeah.
So then one could
You can
do some modeling, right? For your earlier detection,
Yes.
sort of the risk, so that it goes beyond
Yes.
what we're doing today. Yeah, it'd be fascinating.
Yeah. So, in my view, the benefit of overtreatment, the detriment of overtreatment for CAD is minuscule. So if you can improve risk prediction in any way, statins are safe. Lifestyle counseling is safe. Maybe there's a problem with aspirin in a subset of individuals that are older, and we have to be more careful. But those two first things that can reduce risk by 50%, even if you tell someone accidentally, your risk is higher, maybe there's a psychological part that we're not considering, but from the point of view of detriment of overtreatment, there is none. So I consider any improvement as good enough to be used. But there's a lot of people who don't agree with that. It's AUCs and clinical trials. You know, you need that to prove clinical utility. I just don't want to wait for all those trials to finish first before we start introducing genomic data in the clinic. Sorry.
Well, I say that every day. Everybody should be on a statin, but more to the point, I think all of us doing this are a bit disappointed that we haven't been able, despite 600 steps, 600 million steps, we haven't been able to predict this really high proportion of the heritability of coronary disease. So we talked a little bit last night about what we're missing in terms of methylation sites, microRNA, which I think is important, even though we haven't been able to measure it very much. What do you think the next stage is in terms of getting a more omics integration and going beyond the single nucleotide polymorphism?
I mean, I think we talked about this a little bit yesterday, but so I don't see it as a competition. I think in most published studies to date that are looking at other omics, proteomics, methylation, metabolomics, I think you'll see there may be a comparison, and there might be like, a claim that one omics does better than genetics, but they're each providing independent sort of additional risk prediction that is useful. I haven't seen a paper say, well, this makes the genetic stuff, useless. So,
and how does this compare to imaging? Because of course, coronary calcium scores, full cardiac CTA. I mean, you treat the disease, not the predicted risk.
So, great question. I don't think the studies have been done in large numbers, but I think basically you're saying, let's introduce CAC, for example, as one imaging estimate of burden of disease and see, does that kick out other variables? Does it kick out the polygenic risk score? Let's introduce CTA angiograms, which have, even more quantification of burden. I suspect that it might make it dilute the magnitude of effect, but it won't disappear. I'm going back to the other thing is even if it's diluted for CAD, genomics are key for so many other issues. It's we still generate the data, and if we have another test that's not as expensive and better, imaging is pretty expensive. I think I was telling you yesterday about some of the proteomic surrogates for DEXA, and if we can find that's more easily accessible too. If we can find that sort of, you find the proteomic surrogates, then we don't necessarily have to do the imaging, and we get the same information.
Excellent. So I'm wondering if there has been an integration of the genetics of clonal hematopoiesis and polygenic risk score. So of course, this is, acquired mutations within elderly populations in blood cells, which are contributing to disease. And you're kind of implying that looking at the polygenic risk scores maybe is predicting earlier disease. So have you seen anything to say we can have maybe the polygenic risk score that actually protects against clonal hematopoiesis potentially, or mitigates? Are we there yet? If not, do you think that maybe it's just looking at two fundamentally different disease processes? One that's younger, lower, slower, one that's maybe later, maybe more risk factor driven in the very basic senses of the way.
I don't think I've seen what you're talking about. I think it's a sort of like an interaction effect between PRS and CHIP. And you're right, the effects of the PRS are weaker in the older onset disease, while it's the opposite for CHIP.
Right.
I do think there's still risk prediction for older onset with PRS, and I do think eventually, if CHIP can be determined when you have whole genome sequencing, right? It's another sort of benefit. So once you generate the data, you could start working with it. Even if the patients don't want to use it yet, at least if they've given you an opportunity to model it and see if it's helpful to do a combination, or I've seen, there is some CHIP papers that look at mechanism, like the subsets that have had specific genetic mutations and epigenetic genes, methylation genes and telomeres and that kind of stuff, but nothing integrated yet. I think they're still started doing a GWAS of CHIP, basically.
Right, exactly, yeah.
And you may have seen recently the sort of unexpected finding of CHIP and Alzheimer's, right?
Yes,
Yeah.
exactly,
So
yes.
I think that we're still going through to try to figure out that the association between CHIP and coronary disease and atherosclerosis seems to be pretty robust.
Right.
And there's good functional data that's
Yeah,
sort of
yeah.
supporting that.
Mhm.
But I haven't seen, calling them is not, you have to do the genome sequencing and then it actually costs money to call it afterwards.
Right, yeah.
We have 100,000 or actually almost 200,000 MVP of whole genome sequences. And then we have a laundry list of things to do with those sequences, including CHIP. And that's because they cost something.
Right, yeah.
Thank you, Tim. It was a fascinating talk, particularly for someone who doesn't know much about this. But I'm very proud, of course, to be the CEO of a place like this with the work that Ruth has been doing, all the researchers. I think being part of the pioneers is very exciting. But I have a question, actually. So now you have like 500-600. So the question is, would you be able at some point to find the genes that are responsible for the formation of the disease, the plaque anatomically, so you can correct it and not just being maybe a marker, or maybe look at genes that are like creating the inflammation that actually accelerate the formation of plaque and thrombus? And how do you correlate that to the patients, the potential gene characteristics of someone who has a trend to make blood clots? With some people have different ways to coagulate or not coagulate. We have some people bleed easily, some make clots easily. So how do you, because this is all part of the coronary artery disease. So I guess you don't have the answer, but is this something that people think about?
Yes, I mean, I think that's part of what I was trying to show at the end. It's like, I think once you have a set of genetic variants for the outcome, you can then take those set of genetic variants and look at how they link to other phenotypes. And that could include coagulation phenotypes. And it's just, that's your probably most powerful way to determine mechanism. And obviously you're going to look at, you should generate data for the phenotypes that make most sense. So but it's not in many cases, it's not automatically available. Like if you want to look at platelet phenotypes or inflammation, and you have to generate that data. But those individuals who have that data should be taking these lists of SNPs and regions and looking to see how they associate with those phenotypes. But you still also need large numbers.
I can imagine that people have some loci that are silent and something happened in the individual's life, maybe stress, and suddenly the gene is expressed, and then you have the disease. I, as a heart surgeon, have operated so many patients that actually were not having any risk factors, and they had started to have coronary artery disease and severe due to stress. I think stress is underestimated risk of coronary artery disease because we can't measure it. And I can't. And I don't know if genetics can help with that.
Well, that's what I was going to say, that I don't want to say that stress is not in there. It's not, the signal is not in there. It's just that we haven't measured stress, so we can't make the connection yet. So if someone has a large scale, I think the other thing about stress is, like, as you say, it's episodic and
we don't know how to measure it. It's so abstract. But I'd like to comment from Alex about the gender. I think you have an obvious model where you have a lot more coronary artery disease in men than women. So I think maybe go back to the initial approach.
Yes.
Thank you. Thank you for it.
Well, I want to thank you, Tim. This has really been a fascinating lecture, and I think really exciting work that you're doing, and look forward to your next Nature Genetics and Nature Medicine publications. Thanks so much. We have a study.